Okay, then of course we're going to be making sure that you take a look at various hypotheses so that you can note exactly how you are going to evaluate it in the way that you do need to evaluate it. Okay, so please make sure that you remember that. So good things to note. So if we think about this idea, we say, okay, we'll take a look at some errors and we'll take a look at some of those experimental results so we know what's going on. And then we'll take a look at how you can identify the causes and the effects of certain diseases and how you can eliminate, because eliminating is sometimes going to be the only way that you can answer a question, and that is quite all right. Okay, absolutely quite all right, because as long as you get to the correct answer, it doesn't really matter exactly how you got to that correct answer. So with that, let's take a look at the idea of the gene frequencies and these Hardy-Weinberg calculations. Now, before we jump into this, let's kind of get some of these ideas pretty clear so that we can see what's going to be going on. So I'm going to give you an example of a population. So Hardy-Weinberg, this is all about being able to predict what's going to be the percentage of a population that's going to have a particular allele, or the percentage of a population is going to have a particular genotype. So with that idea, let's kind of think about this. So let's say that you have this population, and I'm going to completely make this up. This actually is going to violate one of the Hardy-Weinberg principles in that you have to have a large population. But for the sake of this example, we're going to make sure that you know what we have going on here. Okay, so let's take a look at this population. So here we want to think about the idea of the alleles. So the allele is going to be one version of a particular gene, okay? So we want to figure out, okay, what's going to be the percentage of one allele that we have in this given population. So if we think about alleles, so we know that big A is going to be one allele, it's one version of this gene. What's the other allele that we have presented to us in this population? Yes. And by the way, please feel free to put in your answers. Even if someone has already put in your answer, go ahead and put it in there anyway. The more you participate, the more you're going to get out of it. So we look at this and we say, yeah, okay, so we know that big A is going to be one allele, and we know that little a is going to be the other allele. Now remember, whenever it comes to these Hardy-Weinberg equations, we're going to be assuming dominant recessive heredity. Okay, so we think about that, so we say that big A is going to be the dominant allele. Little a is going to be the recessive allele. So if we think about the various equations that we're dealing with, we say to ourselves, okay, well, we have the first Hardy-Weinberg equation. And that first Hardy-Weinberg equation deals with the alleles. So it says, you know what? We have P plus Q is going to be equal to 1. Now, P is going to be representative of which allele? The dominant allele or the recessive allele? Good. Okay, so we know P is going to be representative of the dominant allele, and we know the Q is going to be representative of the recessive allele. Okay, so that's going to be something that we see here. So we're like, okay, P dominant allele, Q recessive allele. So we know that these two, they are going to be equal to 1. So now if we take a look at this population, one of the things that you'll note is that you can see how many dominant alleles that we have in this population. Well, before we get there, how many alleles, period, do we have in this population? Yes, good. Okay, this is going to be a very important distinction for you to be able to make on test day. Good, okay? So we know that we have 10 alleles. So if we want to figure out, well, exactly how many of those alleles are dominant? So you tell me, how many of these alleles are dominant alleles? Yeah, I like it. We know that 7 of these alleles are dominant alleles. We have 1 that we have right here. We have 2, 3, 4, 5, 6, 7. Okay, so we have 7 dominant alleles. So what the Hardy-Weinberg gives us is the frequencies or the percentages for these alleles. So you say, okay, well, we have 7 dominant alleles out of a total of how many alleles? Yes, okay, so like Tom is saying, we know this frequency is going to be 7 out of 10, or it's going to be 0.7, or 70%. Okay, So we can do a couple of things in order to come up with what Q is. We can use the equation. We say, well, P plus Q must be equal to 1. So if P is equal to 0.7, then what must our Q be equal to if we use that first Hardy-Weinberg equation? Excellent. Okay, so we know that this is going to be equal to 0.3. We can also take a look at it. We see that we have 3 recessive alleles that we have out of the 10 recessive alleles that we're dealing with. Okay, good. So now we're going to turn our attention away from the alleles, and we are going to focus on the genotypes that we're going to be presented with in this case. So then we say, okay, well, we have 3 basic genotypes. What are the 3 genotypes that we have in this tiny population? Exactly. Good. Okay, so that's going to be something that's key. So we say that we have the genotypes. We have big A, we have big A, and we have little a. Okay, except now we have a new equation. So here we have our equation, our P squared, plus our 2PQ plus our Q squared is going to be equal to 1. Okay, so we look at that and we say, okay, plus Q squared is going to be equal to 1. Okay. So this is going to be representative of the genotypes. So P squared, what genotype is that going to be representative of? Is that going to be homozygous dominant? Is that going to be homozygous recessive? Or is that going to be heterozygous? So P squared is going to be homozygous dominant. That's going to be the big A, okay? So then we take a look at our 2PQ. And by the way, you can always feel free to take screenshots. So you can do print screen if you're on a PC. If you're on a Mac, you can do Shift Command 4, which is actually better than your Shift Command 3. Okay, so please make sure you note that. So if you take a look at your 2PQ, this is going to be our heterozygous. So that's going to be our big A, little a. Okay, and then if we take a look at our Q squared, what's that going to be representative of? Good. Okay, so that's going to be homozygous recessive. That's going to be little a. Good. Okay, so excellent job. So then you can do that with this same population. You can figure out what P squared is. You can figure out what 2PQ is. You can figure out what Q squared is. Now, before you figure out the numbers, I'm going to tell you right now that the numbers are not going to add up quite the way that they should. Okay, primarily because it's a small population. But the reason that I give this example is so that students can understand what everything is. Because that's one of the things that I find to be the problem with the Hardy-Weinberg equation. People aren't quite sure what everything is. So just so that you know, the numbers aren't quite going to be 100% perfect. But let's take a look at it in terms of the population. So if we're talking about, well, how many genotypes out of the total number of genotypes are P squared, are homozygous dominant, big A, okay? So how many of these genotypes? It's 3 out of 5. Exactly. Good. Okay, so we look at that and we say, okay, this is going to be 3 out of 5, and we know that this is going to be our 3 out of 5. We think about, okay, we have 1/5, that's going to be 20%. 20 times our 3, we know that is going to be what percentage? So, by the way, you always want to be able to easily go between your fractions and your percentages. So if 1/5 is 20%, then 3/5 is going to be 60%. Good. Okay, so that's going to be something that we're looking at. Good. Okay, so we say, you know what? P squared, that's what that is. Now we take a look at 2PQ. 2PQ, we know is going to be the heterozygous. So what's the fraction that's going to give us the percentage of genotypes that are the heterozygous? It's going to be one of them that's heterozygous out of a total of 5 genotypes. Good, okay? So 2PQ, we know, is going to be equal to the heterozygous. It's 1 out of 5 that's heterozygous. That's going to be 20%. Okay, good. Now Q squared, we can take a look at that also. We say, okay, how many genotypes out of these 5 genotypes are going to be equal to the homozygous recessive? Good. Excellent. Okay. So please make sure that you note that when it comes to these ideas. so that you can be on point. And you'll note all of that is going to add up to 100%. So 60% plus 20% plus 20%, that's going to give us our 100%. So when it comes to these Hardy-Weinberg equations, one of the things that you want to make sure that you do for these questions is identify, well, what are they asking me for? Am I being asked for the frequency of an allele? Am I being asked for the frequency of a recessive allele, a dominant allele? Am I being asked for the frequency of a genotype? Am I being asked for a frequency of a homozygous dominant genotype? Am I being asked for the frequency of a heterozygous genotype? What exactly am I being asked for? And what variables have I been given? So if you want to take a screenshot of this page, you can, because that's going to play a huge role in making sure that you understand these next couple of questions. So I'm going to show you a traditional way to do the questions, and then I'm going to show you a non-traditional way to do some of these questions that'll save you time on test day. Okay, so that's going to be key. So exactly, Tom. The first equation is for alleles, and the second equation is for the genotypes. Good, yes, so remember that. Okay, so hopefully you've gotten a screenshot of that. So let's take a look at a question. So here we have, taking a look at the question, we're assessing it. We see that all the answer choices are going to be numbers. Okay. 96% of the individuals have at least one copy of the dominant normal allele. Okay. So I want you to tell me, what are we being presented with in that first portion? Are we being presented with an allele percentage, or are we being presented with a genotype? If we're like 96% of the individuals, especially when they say have at least one copy, that's going to be kind of the clue there. So when they say at least one copy, that means they can have one copy or they can have two copies. So if we can have one copy or we can have two copies, I'll ask again, are we talking about an allele percentage or are we talking about a genotype percentage? If there's a possibility that we could have two copies of this dominance. So if we're thinking about this idea, the fact that we can have two copies, we can either have big A, we can have one big A, or we can have big A, big A. So that means, what are the two genotypes that are possible? Let's say that we're using A as our alleles. What are the two genotypes that are possible if you have at least one copy of the dominant one? Excellent. Okay, so if you have at least one copy of the dominant one, you know that you're going to be talking about big A, or you're going to be talking about big A, little a. Are these alleles or are these genotypes once you put in big A, or big A, little a? Yes. So like Patricia is saying, these are genotypes. And that's going to be something that is very key towards getting these questions correct on test day. Okay? I just wanted to make sure it was clear. That's all. Okay? You have to understand what you've been given. So we're like, okay. So they said, you know what? 96% of these individuals have this genotype. Oh, okay. So 96% of people that are big A, or big A, little a are like that. Oh, okay. So if we're representing big A, what variable from that genotype Hardy-Weinberg equation are we referring to if we're talking about big A, little a? Is that P squared? Is that 2PQ? Is that Q squared? Yes. Okay, so that's going to be 2PQ, like Molly is indicating. That's going to correspond with our big A, little a. Okay. Now, what variables from the second genotype Hardy-Weinberg equation are we referring to when we're thinking about big A, big A? Good. Okay, so we're thinking about our P squared. So we say, oh, okay. So what equation can we come up with based upon the 96%, our 2PQ, and our Q squared? If they say that 96% of the people have big A, little a, or big A, little a, or big A, big A. Keep it coming. I want you to write it in because the more you participate, the better you get at it. Excellent. Okay. So what? Ooh, I like this thinking ahead. Yes. Okay. Excellent. Okay. So we know that 96% of them are going to be 2p squared plus p squared. Okay. 2pq plus q squared. So as Molly indicated and as Tom indicated, excellent. That means that we know what q squared is going to be. Okay. Now before we start figuring out what q squared is going to be, let's figure out what we need to figure out. Okay, so we just took this first portion of this equation, of this question stem, and we made it into an equation. 96% of the individuals in the population have at least one copy of the dominant. Oh, 96% is equal to 2pq plus q squared. Now, what percentage of the population will be carriers of the disorder? Now, as soon as we start talking about somebody being a carrier, are we talking about one single allele, or are we talking about a genotype? As soon as we talk about somebody being a carrier. So we're talking about a genotype, okay? So if you're a carrier, which particular variable in that genotypic Hardy-Weinberg equation is represented the carrier? Is it p squared? Is it 2pq? Or is it going to be q squared? That's going to be the carrier. Ah, excellent. Okay. So now this is going to be one of the keys to the Hardy-Weinberg questions. Have to identify what you're given, and you have to make sure that you identify what you want. Okay. So we think about that. Oh, okay. So we want 2pq. So we're like, okay, we have to figure out what our p is. We have to figure out what our q is. So we say, oh, like you were saying, if 96% is equal to 2pq plus q squared, then 4% must be equal to your q squared. Huh, okay. So 4% is equal to your q squared. Be careful here. What must your q be equal to? So I want you to think about that. In order to get q squared, we had to square a number. So we have to multiply a number by itself, okay? So we say, well, if q squared is 4%, then q must mean we have to multiply 0.2 times 0.2. Good, okay? So we know the q is going to be equal to our 0.2. Okay, and we had 2 decimal points, so that's how we got 0.04. Oh, okay. So now if q is equal to 0.2, what must p be equal to, according to that first Hardy-Weinberg equation, that p plus q is going to be equal to 1? Exactly. So if we know that if q is equal to 0.2, then we know that our p is going to be equal to 0.8. Oh, okay, so we want 2pq. Oh, okay, so if q is equal to 0.2, and we know that p is going to be equal to 0.8, what is our 2pq going to be equal to? Good. So it's 2 times our 0.8 times our 0.2. Good. Okay, so we know that that's how we're going to end up with 32 as our answer choice. Okay. Take home point here, please make sure that you understand what variables that you've been given in the question stem, take that English, translate it to math. And then please make sure that you understand what variables that the question is asking you for, so you can make sure that you're searching for the correct thing. Okay? Let's do another one. This one is going to look a little bit scary, but I promise it's not going to be as scary as it looks. And in this next one, we're going to do a bit of a practice, so we don't have to do a lot of complicated math. Okay, so let's take a look at what's going on. And as always, if you have more questions about it, please feel free to ask us in the private chat. We'll be more than happy to go over it in greater detail with you. So we look at this one and we say, okay, Huntington's disease, autosomal dominant disorder. Now we take a look at the answer choices because we're assessing the question. And we take a look at the mantissa, which is going to be the first number. Let me see, 8, 6.4, 3.2, 1 point. Okay, so they're pretty far spread apart, so we can be a little liberal when it comes to the rounding. Then we take a look at the exponents. Say -1, -3, -7. Okay, let's see what's going on. So we have the prevalence of Huntington's disease is 6.4 cases per 100,000 people. So I want you to do exactly what we did previously. Take that English and put that into a Hardy-Weinberg equation for us. So if you have the disease, are we talking about an allele or are we talking about a genotype here? Because that's going to be very important to note. Yeah, this is definitely going to be a genotype. Okay, so if it's autosomal dominant, what two genotypes are going to lead you to have the disease? There you go. Excellent. Okay, so you know that you're going to have big A and little a that are going to big A, and big A, little a that are going to lead you to have the disease. Okay. Oh, okay. So what they're saying is, so can you give that to me in terms of an equation that involves the Hardy-Weinberg variables? Good. So we know that p squared plus 2pq, that's going to be equal to whatever 6.4 divided by 100,000 is. Okay? So note that. So is that okay how we got that equation? The p squared plus your 2pq is going to be equal to 6.4 divided by 100,000. Now here is where you start using your math sense in order to get to these questions quickly. So you're like 6.4, 64 is the number that we're going to be dealing with. So therefore, if you know it's going to be 6.4 and all this has to add up to 100,000 or it has to add up to 1 or it has to add up to 10 in this case, q squared, what numbers must be involved in what the q squared is if you know 6.4 plus something must add up to that 1? Exactly. Good. Okay. So we know that we must have a 3 and a 6 somewhere in the q squared. Huh. Okay. Just something to keep in mind. Okay. We're doing estimation, so we don't have to spend a lot of time doing the actual calculations. I went to show you to you both ways. So we're like, huh. Now they're asking us, what's the allele frequency for the disease-causing allele? So what variable are we being asked for? A P or a Q? So it is going to be big A. Let's put it in terms of the Hardy-Weinberg. Yes, okay, so we're being asked for our P. So since we're being asked for our P, we say, okay, well, if Q squared is going to have something that involves a 3 and a 6, Q, because remember, we have to multiply something by itself in order to get Q squared. So what number is going to be involved in Q if Q squared has a 3 and a 6? What two numbers do we multiply by themselves in order to come up with a 3 and a 6, in order to come up with 36? Yeah, we know that Q must involve a 6 somehow. Okay, so if Q involves a 6, what must the P be close to if Q involves a 6? Because we know that P plus Q is going to be equal to 1, so the P is going to involve a 4. So what's going to be our answer choice? That is going to be close, Seth, to that first number being a 4. Excellent. Excellent job. Good. Okay, so we can see that, oh, 8, no. 6.4, no. 3.2? Yeah, that's pretty close to exactly where we want it to be. Good, okay? So that's a nice shortcut. You can do it another way. Another way is going to be correct, but sometimes it can be a little complicated. But if you have great number sense and you truly understand what's going on, you can get to this question very easily and quickly. Okay? So hopefully you have gotten a chance to get a better idea of what's going on with Hardy-Weinberg. So now, let's switch gears a little bit. And by the way, you can take a screenshot of that page if you'd like, so that you can keep it for your records. Let's switch gears a little bit and let's take a look at our second learning goal, where we're going to be taking a look at the various hypotheses that you're going to be seeing in a given passage for testing. So if we think about this, we want to make sure that we clearly understand what the author is saying and how the author can help us answer a question. So this was something that you saw in your full length number 3. So if you remember, this was the passage that was about the pythons. And we had the pythons that would nest in warm environments, and we'd have the pythons that would nest in not so warm environments. So if we look at this, we say, okay, well, the first paragraph basically indicated to us that we have two particular type of pythons. So one of two categories. We have R-selected and we have K-selected. So when we take a look at these categories, we say, well, what's the difference? R-selected reproduce quickly, note that you'll note, and they have multiple offspring at once, okay? Little or no care for their offspring. Okay, so R, very, very quick, very, very rapid. Now, if you take a look at the K-selected, you want to know what those are about. So they're the opposite. So few, large, well-developed young that they pay a lot of attention to, okay? So R, one of the ways that you can think about this, and one of the ways I put it in my head, I said R for rapid. Okay, so R is multiple offspring, they don't really care. R for rapid. And K, they're in it for the long haul with few offspring. Okay, so that makes sense. So nice, because you know you're going to have to differentiate at some point in time. So you take a look at this particular passage, and when you assess it, you note that you have multiple hypotheses that are going to be presented. So you're going to have to figure out the similarities and the differences between these hypotheses. Okay, so it's funny, Tom. So if you note that, you say, okay, I got the difference between them. So then we take a look at paragraph number 2. We look at paragraph number 2, and we say, okay, we're going to start to get one hypothesis here. So water python species, minor changes in the test site location influence the reproductive timing. Ah, okay. So they talked about the idea if you have cool nests, they're going to reproduce slowly, and they're going to wait, and they're going to invest a lot of time in their offspring. But if they have hot nests, then they're going to reproduce quickly, and they're just going to produce a whole bunch of offspring. Okay, so then we get at the bottom. The scientists are going to be disagreeing at how to categorize these. So we're like, ooh, this is going to be the main point of what we're going to be looking at. So if we think about that idea, we say, okay, let's see how they're going to categorize them, because we see that they are going to be disagreeing about how to categorize them. Okay, so let's take a look at the next hypothesis, the first hypothesis. So right here off the bat, you should have read that, you know what? This person wants to classify them as two different subspecies. So the K-selected, that's one subspecies, and then the R-selected for the rapid, that's going to be another subspecies that we're looking at. Okay, and then the author continues to give us evidence. Reproduce slowly over time, the warmness pythons, blah, 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 blah. Characteristics of the K and the R. Okay. So I'm not going over this in great detail because you've already read this passage. What I'm doing is highlighting what you should have picked out when you were going through this on your own. Okay, so hypothesis 1, 2 subspecies. Now hypothesis 2, we know has to be a little bit different. Now remember, the two hypotheses, they're never very extremely different from one another. They tend to be just shades of one another. They tend to have like a Venn diagram of overlap in terms of what the two authors believe. So we look at that. So we say, okay, what's going to be the overlap here? Okay, cannot be categorized either as R-selected or K-selected. Okay, so this second author doesn't really want us to separate them into distinct categories. The author wants us to say, you know what, well, maybe it's harder to figure out which is which. Evidence suggests the author is giving us the proof as to why he or she thinks that. So both cool and warm nest pythons exhibit characteristics of each population category. Okay. So then they start talking about other things that may influence how quickly or how slowly that they're going to be reproducing. So availability of food sources like rats, those are going to influence whether or not they're going to reproduce quickly or whether or not they're going to reproduce slowly. So maybe we can't just separate them into two different subspecies based upon whether or not they are K-selected in terms of them being slow or they are selected in terms of them being rapid. Maybe there's a bit of overlap, and that's exactly what this second hypothesis is going to be all about. Okay, so maybe the environment has effect on it. Okay, so with that, we think about, well, how can we utilize these ideas in order to attack these questions that you'll have to attack on testing? So we look at this and we see, okay. Before the recent Python studies, go ahead and read the question stem and give me a thumbs up when you're done rereading the question stem. You can give me a 6 or a B for a thumbs up. Okay, so we take a look at this, and we have the idea of the amount of time the mother spends guarding the nest and raising her young did not vary across python species. So if we take a look at the answer choices, we're assessing this question using our Kaplan method. We have hypothesis 1, hypothesis 2, neither 1 and 2, both 1 and 2. Okay, so we have to figure out what's going on with the first hypothesis. So the first hypothesis says, you know what, separate them into different subspecies and then base it upon whether or not they're K or whether or not they're R-type. And they have different activities. Okay, some are warm, some are going to be cold nest. So would the author 1 or hypothesis 1 agree that the amount of time in the nest does not vary across pythons? If author 1 is saying, let's separate them into two different ones, because they do change. Would author 1 agree with that? Author 1 absolutely does not support the idea that the amount of time the mother spends guarding her nest does not vary across python species. Author 1 is saying absolutely it varies across python species. Okay, so that's going to be key. So we can eliminate some answer choices. So we can eliminate answer choice A, and we can also eliminate answer choice C. We can't say that it's going to be both hypothesis 1 and hypothesis 2 if we've already eliminated hypothesis 1. Okay, so let's see what's going on. So hypothesis 1 only, cross out the window. So then we take a look at hypothesis 2, and we say, okay, well, hypothesis 2 said maternal nest attendance, it can depend on a bunch of things. It can depend upon the idea of how much food is nearby. It can depend upon the temperature. It can depend upon a whole bunch of things in the environment. Okay, so if we think about what this question stem is saying, the amount of time a mother spends guarding the nest and raising her young does not vary across python species. Does that go with hypothesis 2's idea that, you know what, the nesting behaviors are going to vary based upon a wide variety of factors? So like the food source, the food supply, and the environment in general. Okay, so if we take a look at it, our hypothesis 2 is saying, you know what, let's not put them in different species. Hypothesis 2 says, you know what, they're the same type of snake. They just do different things based upon what the different environment is going to be. So if hypothesis 2 is saying they're the same species, they just do different things, does that go with the belief that it doesn't vary across python species? So it won't necessarily go with that particular belief, because the problem is vary across python species. Okay, so if we're taking a look at that, does not vary across python species, the author for hypothesis 2 is saying let's combine them. Let's put them in the same category. And when we put them in the same category, we'll notice that there are differences within that same category. Okay, so if we find that it doesn't vary across species, that has no finding on author 2's idea that it's the same species. Does that make sense? That if we find that it doesn't vary, if we find that it did vary among species, that wouldn't affect author 2 either, because author 2 is saying they're the same species. So whether it does or whether it doesn't vary among species, irrelevant to author 2, because author 2 is saying they're the same species. They just do different things based upon the environment. So that's how we can come up with our correct answer of D. Okay? So this is a little bit tricky. So that's something that you have to be very careful with when it comes to test day, that you can figure out the nuances of what you have going on. Okay? So let's take a look at another question. So the main difference between the two hypotheses stated above is that. So now we'll have to put these hypotheses in our heads. So we say, okay, hypothesis 1, that they can be different subspecies, but they do different things. Hypothesis 2, that it's the same species. It just does different things based upon the environment. Okay, so we're going to lock that away in our heads so that we can figure out what we have going on. So we say, pretend you didn't see that. Hypothesis 1 suggests that cool nest and warm nest pythons cannot be considered the same species. Okay, so we're okay with that so far. Hypothesis 1 wants to put them in different subspecies, while hypothesis 2 suggests that because both types of python have the same food sources. Okay, this is where we started to go left, because hypothesis 2 isn't focusing on them having the same food sources. Hypothesis 2 is focusing on them doing slightly different things based upon different things in their environment. So we're like, we like the first part, but we don't like the second part. Okay, let's take a look at B. Remember, you don't have to understand every single thing to get to the correct answer. Hypothesis 1, if true, would explain how temperature differences in embryos could affect maturation. What? What type of incorrect answer choice is this? As soon as we're like, temperature differences in embryos could affect maturation. Did we talk about how the temperature differences in the embryos could affect the maturation? It's definitely out of scope. So answer choice B, you are dead to us. We want nothing to do with you. Okay? So let's take a look at C, out of scope, like Brianna and Molly are saying. It was. So C. Hypothesis 1 suggests that cooler temperatures allow the formation of K-selected. Warmer temperatures allow the formation of R-selected. That's okay so far. Hypothesis 2 contradicts this idea. Ooh, that's a little distorting. We don't like that quite as much because hypothesis 2 doesn't contradict the idea. There's just a bit of nuance in there. What's the bit of nuance that hypothesis 2 added in? I like what you're saying, Molly. Yeah. So here's the detail, a little bit faulty. The nuance that hypothesis 2 added in was that you have a whole bunch of other factors that are going to play a role in this, okay? So it's not only the temperature, it's going to be the food supply, it's going to be other aspects of the environment. So that's how we can get rid of C as the answer choice. And you know what? As a good Kaplan student, once you have taken the time to wade through these answers, because it's taking some time, you're going to pick D and you're going to move on. You're not going to waste time. Okay? So you're done. D for done. Good. So excellent job. So please get used to the idea that when you have different hypotheses, that you'll see a wide variety of different nuances in the two hypotheses. And it's going to be up to you to weed out those extreme answer choices or those distorted answer choices. Okay? So go ahead and use those ideas that we talked about to review the rest of the questions that are associated with that passage. And yes, Tom, you would definitely triage this question. You absolutely would, because you know it's going to take you some time. Okay? So let's take a look at learning goal 3. So we'll take a look at an experimental passage. So some of the passages that are really hard are experimental passages. So we'll make sure that we can see what's going on. So this passage should be familiar to you. If you remember, this was the passage about the radioimmunoassays. So we look at it and we say, okay, well, let's take a look at what we should have noticed from this passage. So here we're trying to measure the concentrations of a hormone in the blood. Okay, so here we have we're using antibodies in order to do that. So what we're going to be doing, according to this passage, is we're going to inject a portion of, let's say, the organ that this hormone is made in into an animal like a rabbit. So we're going to inject a portion of like the human liver or something into a rabbit. And the rabbit is going to be like, that is not rabbit tissue. That is human tissue, and therefore what's the rabbit immediately going to do? What's the rabbit immediately going to make once this rabbit is injected with this human tissue? That are going to mark this tissue as human and mark it for destruction. Exactly. Okay, so the rabbit is immediately going to make antibodies to this. Okay. So what we can do with these antibodies is we can label it. Okay, so that's one of the things that we'll know. So we take a look at this and we say, okay, so here we have the antibodies, and we can label some of the hormone of interest. So we label some of the hormone of interest. And then we make sure that we make, if we take a look at the blood sample that we're going to assay, the blood sample that we're going to assay, it's going to have that same hormone in there, but it's not going to be labeled. Okay, so we have antibodies, we have unlabeled hormones, and we have labeled hormones. Okay, well, the labeled hormones and the unlabeled hormones, they're still going to be competing. They're going to be competing for that antibody. Okay, now we'll see a diagram of this on the next page. So they're competing for that antibody. Okay, so let's see what's going to happen. So what we do is, since they're competing for the antibody, the more of the unlabeled hormone that's going to be in the blood supply. So we want to think about this idea. So the more of the unlabeled hormone that's going to be in this blood supply, we want to think about what exactly is going to be happening. So we say, okay, well, based on this idea, and we'll go back to the slides that are going to be necessary. Every once in a while, the slides do get a mind of their own. So we think about this, and we say, okay, so here you have antibody, labeled hormone, and unlabeled hormone. And they're both competing for the same antibodies. If you have a whole bunch of unlabeled hormone, are you going to have a lot of the labeled hormone binding to the antibody? They're competing. If you have a whole bunch of unlabeled hormone, are you going to have a lot of the labeled hormone binding to the antibody? No. Okay, so that's the main idea. And the opposite is going to be true also. So if you only have a little bit of unlabeled hormone in the sample, what's going to be the amount of labeled binding to the antibody? Is it going to be high or is it going to be low? If we only have a little bit of unlabeled and a lot of labeled, is there going to be a lot of labeled binding to the antibody or a little bit of labeled binding to the antibody? Okay. So I want you to note it's the opposite. If we have a little bit of hormone, of unlabeled hormone, that means that more of the labeled hormone can bind to the antibody. So let's review that. Unlabeled, labeled, they're competing. A little bit of the unlabeled, more labeled can bind. A large amount of the unlabeled, less labeled can bind. Does that make sense? They're competing. A little bit of unlabeled, a lot of labeled binds. Large amount of unlabeled, little bit of the labeled is going to combine, is going to bind. Okay? So kind of roll that around in your head as the main idea that we're dealing with here. Yeah, okay, cool. I'm glad we're all on the same page. Good. Okay? So that's going to be the main point that you get from this particular passage, because you have to make sure that you clearly understand what's going on. Okay? And then they generated a standard curve. And they said, you know what? We use the standard curve so that we can figure out how much of the hormone is going to be there. So it's really important to get these ideas, like Eli is saying, before you move on to the questions. Okay, so here we have the curve. And you'll note exactly what we just said. So if you have the labeled hormone, if you have a lot of the, and even if you didn't get that, if something's important, the author is going to repeat it, okay? So if you have a large amount of labeled, because we're going up this y-axis, that means that you have a small amount of the unlabeled hormone in the blood. So tell me what D represents right here. High labeled or low labeled? We have warring answer choices. So the labeled, which axis is it on? Is it on the y-axis or the x-axis in this diagram? So labeled is on the y-axis, okay? The concentration of labeled is on the y-axis. So D, is that a little bit of labeled hormone bound, or is that a lot of labeled hormone bound? Yes, okay, so we can see that D is going to be a small amount of labeled hormone bound because it's going to be low on the y-axis. Ah, but if we have a small amount of labeled hormone that's going to be bound, what does that mean about the amount of unlabeled hormone? And we can take a look specifically for D. Do we have a high or a low amount of the unlabeled hormone that we see on the x-axis? Exactly. We have a large amount of the unlabeled, so we can figure it out. Good, okay? So that's something that was presented to you both in text and on the diagram. And that's a relationship that you want to make sure that you understand for testing. So good, okay? So we look at that and we say, okay, the two forms are similar in structure and chemistry. They said that the form that circulates in the blood, the active form, is different from that extracted from the tissues. Okay, that's good to know. Active form may be different from that extracted from the tissues. Okay, so good deal. And you can put a label like that on this paragraph. So with that, let's take a look at what's going on. So this is going to be the idea that we talked about, that we injected the mouse, it gave us antibodies, we labeled some hormone, we didn't label other hormone. Okay, the ones that more labeled, that means that less unlabeled, and vice versa. So if you want to take a screenshot of this page, you definitely can, because it's a beautiful schematic about exactly what we just talked about. You can do your print screen, or you can do your Shift Command 4, which is better than your Shift Command 3, in order to get this nice schematic out. Okay. So let's use these ideas to tackle the questions. So we say, okay, well, based upon what we have, let's take a look at 48. So here they're taking a look at the idea of the principle that we just talked about. Radiolabeled and unlabeled hormone is going to compete for binding sites, which is not going to compromise the validity of this. So if labeled and unlabeled have to compete, what's not going to compromise the validity? This is one that you'd probably want to triage for later because you'd have to go through each answer choice piece by piece. But let's do it now. Let's see. The antibody binds the radiolabeled hormone with a greater affinity than unlabeled. Would that compromise the validity of the RIA of this assay if one has a greater affinity for the antibody than the others? Absolutely. Absolutely. We want nothing to do with answer choice A. No, 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 no. Nothing to do with this. You're dead to us. Okay, let's see. B. The antibody binds the radiolabeled hormone and the unlabeled with equal affinity. So that compromises the validity if they're bound with equal affinity? No, it absolutely would not. And we like this question because Tom, yes, Tom, yes, 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 yes. That's exactly what I was about to say. Pick B and move on, okay? We had two diametrically opposing answer choices. And we said, you know what? B is our answer. We're not even going to bother with the rest of them. Okay, excellent, good. Okay. So now let's take a look at another question that's based upon this idea, because we've talked about how we get to this answer choice. Okay. Let's see here. The standard curve for insulin, if this figure is that, okay, would most likely represent one before glucose infusion and after glucose infusion. Okay, so let's take a look at what's going on. So before glucose, and then we have the idea of after glucose. Okay, so this is kind of testing us a little bit on our knowledge of our hormones. So what should we expect to happen to insulin levels before we have glucose? Low insulin levels. Good. Excellent. Okay. And then an hour after glucose, what do we expect to happen to insulin levels? Yeah, we expect them to be high. So we want an answer choice that goes with that. So look at this. Even if you had no idea what was going on in this passage, there you go, Tom. Excellent. If you have no idea what's going on in this passage, you can say, you know what? We can remove any answer choices that do not show a high extreme and a low extreme, even if we don't know what the high extreme is, or even if we don't know what the low extreme is. Okay? So if we have something that said B and C, we're like, what's one answer choice that we're going to be like, okay, goodbye, please. Exactly. So even if we have no idea about what's going on, we can eliminate C and C. So we're like B and D. What's another one that we're going to eliminate, because they're pretty close to one another, and we want something that's one that clearly says low and one that clearly says high. So you'll note, yes, exactly. A and B, we're like, we want something that's clearly high and clearly low. So now we're down between B and D, or we're down between C and A. So let's see if we know what's going on. What we're measuring, that's going to be the unlabeled hormone. So remember that, okay? What we're measuring is going to be this unlabeled insulin. So we want initially the unlabeled insulin to be low. So is it going to be the left side of the graph or the right side of the graph that shows low unlabeled insulin? Because I want you to note that the x-axis is that unlabeled insulin. So which one is going to show us low unlabeled insulin? So here we have low x-axis, medium x-axis, and then high x-axis, which is high insulin. So which side is going to be the low unlabeled insulin? The left side or the right side, if we're following along this x-axis? There we go. The left side is going to be low insulin. So we're like, you know what? We want an answer choice that starts off with one of these initial points that we're going to be dealing with. So we're like, oh, okay. Well, what answer choice is going to go with that idea? We see that our B and our D is going to go with that idea. And we actually didn't have to sit there and figure out every single thing that we had going on. Okay, so that's going to be something that is key towards making sure that you know how to get to the correct answer on testing. Okay, so let's do our last learning goal, and you can take a screenshot of this page if you want. We're going to do a nice question that is going to go with this main idea, and you can do number 50 on your own. So we have to take a look at the idea of the effects of various diseases and see if we can identify them based upon the question that we're given. Okay, so let's see what we are dealing with. So we say, you know what? Do question number 16 for homework. Let's do the next question. That's another nice one also. Number 58. Nucleoli in the alpha cells will be the largest in a patient with. Okay, alpha cells, what are we talking about? Especially if we take a look at the answer choices and we see diabetes, pancreatic cancer, acute pancreatitis. So we look at that, excellent. Okay, so we know that we're talking about the alpha cells in the pancreas are going to be making the glucagon. Okay, so we're like, okay, in what case? Now note, it said the nucleoli. What do the nucleoli make? Can you remind me? Yeah, these are fast points. So be careful here. It's not the nucleus. It's the nucleoli. So what is it going to be partially responsible for the production of? Okay. I see we have a prediction already. So one of the things that you'll note here is that we are going to get tons and tons and tons of our protein. We're going to get tons and tons of our glucagon because the nucleoli are going to be responsible for producing those ribosomes. There you go, Molly. So that means tons and tons of protein. Which one of these answer choices goes with the idea of tons and tons and tons? Too much production if you have a very, very large nucleoli that's producing lots of protein just completely out of the, yes. So that's how we know that our correct answer is going to be cancer. Okay. So excellent job in terms of making sure that you can get to the correct answer even without fully understanding everything. So please use that idea in order to tackle these types of questions on testing. Okay? Good deal. So with that, thank you very much for attending. We definitely appreciate it. Please make sure that you can tackle these Hardy-Weinbergs the way that we did. Note the nuances of the hypotheses when they're different. And please make sure you can note the experiments, how to understand them, and making sure that you know how to identify diseases and symptoms. Okay? I wish you the best of luck on your MCAT, and I'll see you next time. Bye.